Hi dear students, a warm good morning to all of you and welcome to one more session on biomolecules. In the previous sessions, I had discussed on you what is DNA, what are the different types of RNA, and today we are going to talk about something else. I'm not going into the heading of the topic, and I guess from the figure itself, you will get an idea what are we going to talk about. Yes, in the picture which we see here, you can see you have a double-stranded DNA, and at one particular position, the double-stranded DNA is being unbound to form single-stranded DNA, and onto one particular strand, there is attached what? A particular enzyme that is called the RNA polymerase. And using the enzyme RNA polymerase, you can see the synthesis of something happens. And what is that strand here? The additional strand which has been formed here is the RNA. So, what are we depicting or what are we going to discuss? Yes, we are going to talk about the transcription of RNA. That is, the process of conversion of DNA to RNA with the help of the enzyme RNA polymerase is called the transcription. And in the coming sessions, we are going to learn on the various aspects of transcription. Now, transcription, as we all know, both in prokaryotes and eukaryotes, they require the conversion of DNA to RNA and they require RNA polymerase. But there do exist some differences between the prokaryotic transcription as well as the eukaryotic transcription. So let me just start first with the transcription in prokaryotes. As I mentioned earlier, transcription is a process of conversion of double-stranded DNA into the formation of RNA. And prokaryotic transcription, as I am explaining right now, it mainly happens within the cytoplasm of the organism, because we know that in the case of prokaryotes, they do not have a true nucleus. And so the process of transcription or the process of conversion of DNA to RNA mainly happens within the cytoplasm. And the process of transcription is immediately followed with the process of translation or the protein synthesis. So the prokaryotic transcription as well as the prokaryotic translation happens alongside within the cytoplasm. And RNA polymerase is the enzyme which is being utilized for this process. And you can see that it does not require any additional enzymes like primase or anything. In DNA replication and all that, sometimes we will need additional primers for the formation of additional DNA strands. But here, when an RNA is being synthesized from a DNA, the use of primers as well as primase enzymes do not occur in the process of transcription. And the building blocks which we use to produce RNA, they are ATP, GTP, CTP and UTP. Of course, in DNA, you know, it is a thymine which is being used. This is the TTP. But here, instead of thymine, we have the uracil or the UTP. Uracil based UTP is being used as a building block to produce an RNA which is complementary to the DNA template. And this mainly happens within the 5-3' direction with nucleotides being added to the 3' end of the growing chain. Now let's talk about the main enzyme which is being involved in the process of transcription. So I am concentrating mainly on the prokaryotic transcription. If I go to consider the prokaryotic RNA polymerase, it mainly consists of different subunits, the alpha subunit, the beta subunits, the beta dash subunit as well as the sigma factor. So you have different subunits here, and there are mainly two alpha subunits and two alpha subunits, beta dash as well as there is a sigma factor, as well as you also have another one called the omega factor. Now the sigma factor is a one which has no catalytic activity, but mainly it helps the core enzyme to recognize the start of a particular gene. And when the sigma is being bound to the core enzyme, the 6 subunit complex is called the RNA holo enzyme. And only the holo enzyme can begin the transcription, but the core enzyme completes the RNA synthesis once it has been initiated. So if you go to look, you have what? Different subunits, as I told you, and different subunits have different roles. So when the sigma factor, so now the core enzyme, it is mainly consisting of alpha to beta, two alpha subunits, beta, beta dash, and the omega one. But only when the sigma factor comes and binds to it, the enzyme, the core enzyme becomes complete and becomes a holo enzyme. So you can just see, you can just imagine like this. Unless and until the sigma factor is being bound onto the RNA polymerase, the RNA polymerase will be just slide past through the DNA. And when the sigma factor comes and binds onto the RNA polymerase completely, what happens? The RNA polymerase is able to identify the region or the start from where the transcription or the starting site for the transcription to happen. So, if the sigma factor is not coming in binding, the binding of the RNA polymerase or onto the onto the DNA, so that it synthesizes specific RNA, will not happen. Okay, so let's move on to that. But the precise function of alpha, beta, beta dash, and omega, it is not yet clear. But we say that alpha subunits play the chain initiation and interaction with regulator proteins, and beta subunits play a role in the chain initiation and elongation, and beta dash help in DNA binding. But the recognition of the promoter site or the site from which the RNA has to be synthesized, that is mainly being done by the sigma factor. So, if you're going to take a prokaryotic gene structure, what happens? That is, gene, of course, is a part of DNA. So, if the gene has a start site, now this is the transcription initiation site is a site from which the RNA has to be synthesized. And you know that is the what region, that RNA is the one which will further code for a particular protein. So if you go to take a prokaryotic gene structure, it will have a transcription initiation site, and ahead of that there would be a particular region called the what? Promoter region. And the promoter region is being found to be upstream or towards a 5' end of the gene start. And towards the 3' end, there is also another region called the terminator sequence, or it is called the downstream sequence. So if you go to take any DNA of a particular prokaryote, okay, when at the region where the gene is starting, just before that, there will be a site called the promoter sequence uh, ahead of the transcription initiation site. And after the transcription has to be stopped, like after the gene is over, after the RNA coding sequence is over, there will be a transcription termination site also. And what is this particular promoter and what is its significance? A promoter is a sequence of DNA to which the proteins bind and that will initiate the transcription of a single RNA from the DNA downstream of it. And bacteria, if you go to take this promoter sequence of bacteria, you can see that it is having two sequences which are very much of importance. One is at the -10 position and one is at the -35 position. Uh, Often TT Gaza, which is being found at the -30, about 35 base pairs before the transcription starting point. And we also have a Tata box or the Pribnow box, which is being found at the -10 sites respectively. Now you will think, what is this -10 and -35? Now if you go to consider this as the transcription initiation site, if you move back -10 base pairs back, then you find the what? Tata box. And minus from this transcription initiation site, if you move backwards 35 base pairs, then you find the what? You have the what? TT Gaza site. So these two sites are being found in almost most of the prokaryotic promoters. And the RNA polymerase holoenzyme recognizes the specific sequences at the -10 and -35 sites of the promoters. And because they are similar in all promoters, they are called the consensus sequences. And once the bound to the promoter site, the RNA polymerases is able to unwind the DNA without the help of any helicases. And the -10 site is rich in adenine and thymines. And you know that if there is a AT bond, it is very much easier to break the hydrogen bonds. So this is and it will be easier to unwind the DNA at the data box. So at the -10 we have the what? -10 we have the Tata box or the Pribnov box, and at -35 we have the what? TT Gaza site. And the -10 site is rich in adenine and thymine, as I told you. So it is very easy and you will get an open complex. And the region of unwinding is called the transcription bubble. So once the DNA, once the promoter is being formed, promoter region has been identified, what the unwinding of DNA is been happening ahead of the transcription initiation site. Now, what are the steps of transcription? First, you have the initiation, elongation, as well as the termination. Now, if this is a gene start site, transcription start site, ahead of that you have the promoter sequence, the -10 sequence with the Tata box, and the -35 with the TT Gaza site. So, one is a coding strand and the other one is a template strand. So, you look, the strand with the 5-3- end is considered to be as a coding strand. And as I told you, RNA transcription or the formation of RNA happens only in the 5-3- direction. So the transcription or the formation of RNA will happen only from the template strand, which is in the 3- to the 5- end. So in the process of initiation, you can see that only a relatively short segment is transcribed, and the initiation begins when RNA polymerase will bind to the what? It will bind to the promoter for the gene. And RNA polymerase coenzyme is not able to bind DNA tightly or specifically. Only when the sigma factor comes and binds to the core enzyme, it becomes a hollow enzyme and which binds the promoter tightly. And the promoter serves only as a target for the binding site of the RNA polymerase, and that is not being transcribed. Now you should have a look at this particular figure. When the transcription is starting, the enzyme is coming and binding onto the promoter region, but the region of the promoter region is not getting transcribed. That is, the DNA in this promoter region is not getting transcribed. It is only the region where the the RNA polymerase comes and bind. The transcription starts ahead of that, at the transcription start point. So we are going to the initiation. And in the case of bacteria, the sigma factor of the RNA polymerase holoenzyme, it is that is responsible for the positioning of the core enzyme and the promoter. And the sigma factor recognizes the two regions of the promoter, one centered at the -35 and the other centered at the -10. And once positioned properly, the DNA at the -10 region unbinds to form an open complex, and the sigma factor dissociates from the core enzyme after the transcription is being initiated. Now the next step what we have is a elongation. The transcription bubble will move with the RNA polymerase as it proceeds to transcribe mRNA to the template DNA strand during the process of elongation. Now what do you mean by elongation? Elongation is a process in which what? The RNA is being synthesized and the length of this RNA is being increased. And during this process what happens? There would be a state in which the RNA polymerase is moving ahead of it, ahead of the template. And when it moves as such, what would happen? There would be temporary RNA DNA hybrids being formed, and as this RNA polymerase is progressing, okay, is moving in like what? It is moving on the template in the 3' to 5' direction. Okay, but the RNA is being made in the 5' to 3' direction, and it is complementary to the template DNA. Now you can have a look at this. See, this is a template strand. On the template strand, the RNA polymerase is moving from the 3' to 5' direction, but the synthesis of RNA happens within the 5' to the 3' direction. So as the elongation is being proceed, what happens? You will get a single stranded mRNA, and this mRNA is being released, and the two strands of DNA will begin behind the transcription bubble will start winding. So just have a look at this. If you have a region, this is called the transcription bubble, where the double stranded DNA is being unwound and the RNA polymerase is being this is RNA polymerase. As the polymerase is moving, what happens here? RNA is being synthesized, and it mainly happens in the template strand, and RNA synthesis happens in the 5' to 3' direction, and you can find a RNA DNA hybrid which is being formed in between also. And the nucleotides are being added to the what? 3' end of the RNA. And then finally we have the termination step, in which the what? The transcription has to be stopped. And for that, there should be a term, something should say that termination or the transcription has to be stopped. So the end of a gene or a group of genes is marked by a DNA sequence at the end, and that is called the terminator. Okay, and the sequences within the prokaryotic terminators often contains nucleotides which when transcribed into RNA form hydrogen bonds with a single stranded RNA. And this intra-strand base pairing creates a hairpin shaped loop and structure. We can just go into the details of that. There are mainly two kinds of terminators. One is the intrinsic or the raw independent terminator, and the other one is a raw dependent termination. So we'll just have a look at that, both of that. What are they? In the case of raw independent terminator, it is an intrinsic termination of RNA, and what happens? Hairpin-like structures will be formed, and once the RNA polymerase are paused at the hairpin loop structure, the AU base pairs in the uracil region are too weak to hold the RNA DNA duplex together, and the RNA polymerase falls off. And the intrinsic termination is when the RNA forms a hairpin-like structure and which displaces the RNA polymerase and stops the transcription. Whereas in the raw dependent termination, which is the second kind of terminator, it does not have a poly-U region, and often the hairpin is also it lacks the hairpin as well as the poly-U region. And so in such cases, what happens? Specialized proteins called the raw factors will come and bind, and the terminator causes a raw dependent termination, and the raw binds to the mRNA and moves along the molecule until it reaches the RNA polymerase that has halted at a terminator. So the raw factor, as a hybrid of RNA DNA helicase activity, causes the RNA polymerase to dissociate from the mRNA, and the unwinding of the mRNA DNA complex will happen. Now just look into this. If you are having a what? Raw independent termination, just look at this. If there is a what is it? This is an RNA molecule. A RNA molecule is being formed, and what happens is it will form a what? A stem loop structure will be formed upstream 7 to 9 with 7 to 9 uridine residues. So what happens is at that when it reaches such loop-like structures, the RNA polymerase will get paused or it will be stopped for some time. And the uridine-rich sequence in the open complex is not, and when it reaches such uracil-rich residues, what happens? I made a mistake earlier. Uracil-rich residues, what happens? The RNA polymerase is forced to leave the template and the termination will occur. Okay, and so this is a method of termination where uracil-rich sequences are being formed in the complex, and then when uracil-rich residues are being formed, they will form stem loop-like structures, and that will halt the RNA polymerase and stop it over there so that it will be forced not to add further RNA molecules. That's the case of the Rho independent termination, which is being found in the prokaryotes. Now, in the case of Rho dependent termination, you do not have any what happens stem loop like structures, but there are certain proteins called the Rho proteins. These are helicases. They will come and bind onto some sequences called the Rho recognition sequences. And once a Rho protein comes and binds to it, what the RNA polymerase will reach a termination site, and it will be also caused to stop. And as a result, what happens? The termination of transcription will happen. So transcription in the case of bacteria, and I mean by prokaryotes, they mainly has three steps: the initiation, elongation, as well as the termination. And the termination can be formed either in the Rho dependent as well as a Rho independent manner. So I hope this is clear. We will be continuing with the eukaryotic transcription in the next session. Thank you for now.